talk today about is the question of gene flow in species delimitation studies. And so I'm going to try to convince you that if we really want to get at the question of species boundaries, we need to somehow model gene flow and consider it explicitly as we do our investigations. And so before I go any farther, I just want to acknowledge Bryan O'Meara, Nathan Jackson, and Ariadna Morales-Garcia, who have all worked a lot on this project that I'm going to talk about today. So the motivating focal system is a brown bat from North America, Migrationlimitation. And it's a species that has this very wide distribution throughout most of the continent. And the bat is actually under a lot of conservation pressure right now due to white nose syndrome, which is this little fungus like athlete's foot that prevents it from hibernating throughout the temperate winters. And so because they use all of their fat trying to wake up and groom themselves, they end up having to come out of hibernation far too soon and not having any insect seed, and then they die of starvation. And so this species is about to be listed. And the big question in this species has always been, what is the actual taxonomic status of these named and described subspecies? There's at least 4 of these subspecies that are commonly considered as fairly distinct by mammalogists. And a few years ago, along with Tanya Dewey, a long collaborator of mine, we did a species limitation study that looked at Migrationlimitation and several other species. And you can see that each of these has a bunch of named subspecies. And what we wanted to ask was whether or not the species tree model that treated the nominal species as terminal taxa had a higher probability than the one that treated the subspecific taxa as terminal. And as you can see, this species tree that went down to the level of the subspecies had quite a higher probability. And so this suggests, and other species limitation analyses have supported this, that in this clade of bats, it's better to think of these nominal subspecies as being independent lineages. But this is always, so I should say that even though we did this approach a couple of years ago, as new species limitation methods and improvements have been developed, I sort of used these same data to try them out, and in every case, they've supported this idea that these lineages are separate. So we can use BPP, which I think is one of the leading programs for this type of analysis, and we get the same results, very high posterior probabilities on the model of all these lineages being separate. But this has really bothered me in a lot of ways, because when we think about species, particularly for vertebrates, we often have sort of been introduced to this idea in the context of scientists like Ernst Mayr, who really put a high premium on reproductive isolation, the role that gene flow plays in keeping populations connected. And none of the species trees analyses that form the basis for these newer coalescent-based species limitation methods actively consider gene flow in any way. They treat gene flow as not part of the model that's underlying the analysis. And in all of these species tree methods, there's a number of assumptions. But what they all share is this idea that anytime you observe shared polymorphism across different lineages, this shared polymorphism is a remnant of some ancestral polymorphism, and it just hasn't sorted due to genetic drift yet. So the idea that this shared polymorphism could result from allele sharing as a result of migration and gene flow isn't explicitly part of these models. And of course, we know that gene flow is something that, in some cases, can interfere with our ability to use species trees to estimate phylogeny well. There's a very good paper that just was published in Systematic Biology last year by Adam Lachey and co-authors, where they do an extensive study looking at how and why gene flow interferes with the ability of species tree methods to estimate the phylogeny. And they show that in a lot of cases it does have an effect. Okay, so if gene flow can influence our ability to estimate species trees well, and we're using all of these species tree-based methods for species limitation, to me this suggests that it's possible that the presence of gene flow is something we should be worried about and concerned with if we're seeking to delimit species. And this is a hard question to get at, but it's something that research in my lab has been working on for a number of years. And I think particularly in systems like the one that we're working on here. So here's Ariadna collecting a bat. Myotis bats are volant. They fly very well. They can fly very long distances every night. And the ranges of these subspecies are contiguous across broad parts of North America. And so to completely ignore the possibility of gene flow in systems like this, to me, seems to be very short-sighted. And I think explicitly looking into gene flow is justified, particularly in cases like this, where you have different groups coming into contact. And so the question of how we detect gene flow, I think, is an important one, and it has a couple of different answers. One answer is maybe straightforward. We use a program like Migrationlimitation that's actually trying to estimate gene flow. And so, as many of you know, Migrationlimitation implements a model that's called an island model. It has different populations, and these populations are connected through the sharing of alleles. You can run this very nice software that Peter Beerley has been developing over the last decade or so, get estimates of population genetic diversity theta, as well as the rate of migration to and from various populations. And we can do that in the meiosis back system, and if we use this full model, what we find is that we can estimate rates of gene flow in the coalescent among all these different entities. And so we get a number, we get a rate of gene flow, but what we don't get is any way to easily know whether these parameter estimates are important enough to treat as, be connecting these populations. Another thing that Migrate does recently, the newer versions, is it allows us to select different models and to identify the particular migration model that's the best fit to the data. So we saw a talk yesterday that did a very nice example of this. We can do something like this in our bats. And what we find is that at least 3 of these named subspecies are well connected by gene flow using the model that's most supported given our data. And so what this suggests here is that we have 2 very different answers depending on how we analyze our data. If we use a species tree model and something like BPP, we would assume that these different described subspecies are all independent, and we should probably think of them as different evolutionary entities. And if we use this island-based migration model, we would have to conclude that at least 3 of these things were well connected and probably should be thought of as the same. And so the question, if you're interested in empirical systems, I think is very clearly which one of these models is best. And the unfortunate thing is it's hard to know because they use different machinery and you can't really directly compare the 2. So the same data, we have different interpretations. And one of the main objectives of research in my lab has been to develop ways to compare very different demographic models and assess the fit of these demographic models to the data that we can collect. And so what I'd like to talk to you today briefly is a method that I've been working on with Bryan O'Mara and Ariadna and Nathan called FRAPL that tries to do demographic model selection using what we call approximate likelihood. And so I'll explain how we calculate these approximate likelihoods in a second, but basically we think of FRAPL as analogous to the program model test, what many of us have used to estimate models of sequence evolution. We're not trying to use FRAPL to be the program that we're going to use for really accurate parameter estimation for sort of the end of the analysis. But what we want to do is see which of the models that we might use to analyze our data is most appropriate for a particular data set. So think of FRAPL like model test. And like model test, we need to do a few calculations to get an idea about which model is the best. So what FRAPL does is it uses observed gene trees from the data. And gene trees are, of course, a way of summarizing phylogeographic data that date back to some of the earliest phylogeographic studies. Many of the talks that we've seen today and yesterday have included gene trees as part of the analysis. And so what we need to start a FRAPL analysis are gene trees, assignments to populations. So here we're using 3 populations, A, B, and C. And the number of parameters that you want to include in your analysis. So the more parameters you have, the more dimensionality the demographic model space has, and maybe the harder it is to choose a bunch of models. And the user can set all of these inputs into the software. And so what FRAPL basically does is for different models of historical demography, we generate simulated data and see which of the simulated data is the best match to the empirical data that we've observed. And so whether it's an isolation only, or a migration only, or an isolation and migration, we can generate distributions of gene trees under models and then see which distribution produces data that are the most like the data we've actually observed. And so to do this, we need a number of parameters, of course, and these parameters are the same that we are used to in other programs like divergence time, migration rate, effective population size. And we go through, and for each model, we generate a very large number of trees. And then to calculate the approximate likelihood of our data given the model, we just count up the number of times that we see a topological pattern that matches what we observe in our empirical data. And so if our observed tree is here and one matches that, then this gives us some idea about the relative probability of the data we've observed given a particular demographic model. Okay? And so one match in 50 gives us an approximated likelihood for this model. We then do this for a number of models, and without getting fancy with math, I could probably convince you that if we have 5 matches under one model and only 1 match under a second model, this first model is a better fit to the data. So it's a very straightforward approach. It uses nice, easy math that I can understand, and I really like that. And so let's get back to the actual cool part of the talk, these bats that we've been interested in for several years. So when we use the same 7 locus data set that we used in my earlier species delimitation analysis, what we find is an isolation model that basically has secondary contact between 2 lineages that are not sister is the best fit to the data. It doesn't have a great model probability, as you can see, so about roughly 40% of the weighted model probability, so this is directly analogous to a relative posterior probability, about 40% is this model. There's 2 other models that have a fairly high support, and then 213 models that are all sort of summed together. The nice thing about FRAPL is you can do very large numbers of models and compare them in a reasonable amount of time. I say reasonable, this took maybe a week on a fairly fast computer. We didn't want to stop with just 7 loci, so Ariadna has collected a very nice data set of ultra-conserved elements and some other captured markers. And she's used an approach that's basically one of these capture arrays that microarray has synthesized. And so we've gone through and done all of that. And we can now use a larger data set to evaluate the same group of models. So for this analysis, I'm just showing you the results from 100 UCE loci. The nice thing about FRAPL is we just do this on a locus by locus basis, and the more data that you have, the greater the chance of observing the same topology is, and sometimes you get these efficiencies because you see similar topologies that you can then compare against all the models. So it actually scales well with larger data sets, and you can do it with these very large data sets, even up to thousands or tens of thousands of loci, particularly if you have a cluster computer. Okay, so the 216 models are a lot of different demographic models that include 4 types. So there's island, divergence only, isolation with migration, secondary contact. And when we calculate the approximate likelihood of all these models and then use information theory to sort them and rank them, what we find is that one model actually has almost all of the weighted model probability. So this model here, and I'll explain what this is in a minute, has over 99% of the total support. This is something we see a lot with FRAPL. As we get past about 75 loci, our ability to discriminate among models gets really good. And so even though 75 loci seem like a lot, when you're using next-gen sequencing methods, it's really not a very large amount of data. And so it appears that we're able to do this, simulations back this up, that we're fairly effective at identifying demographic models that are a good fit to the data. So instead of showing you a pie chart with a bunch of different models, we basically have the whole pie here, right? And this is really nice when you want to know which model is a good fit for your data. So this migration model looks something like this. There's these 4 species, and again, 2 species that aren't sister are sharing alleles, but it's actually different than the model I showed earlier because the topology here is a little different. The topology of the model of the 7 locus data set matches what we see in BEAST, both when we analyze that 7 locus data set and when we analyze the 100 locus UCE data set. So this is an example of gene flow interfering with our ability to estimate the species tree well. And I think it's an indication that we need to think about gene flow more carefully, even if we're looking at systematic questions. The real challenge, though, and the thing that we don't have a good answer for, is what to think about the species status here. So how many people think that we really have 1 species in this system? Okay, some hands. What about 4 species? How many people think we have 4 species? How many people think we have 3 and that these 2 should be the same even though they're not sister taxa? How many people just don't have any idea? And this is a real challenge, but I think until we're honest about how we interrogate our data and we ask the hard questions, it's going to be easy to mislead ourselves and to fail at species limitation because we're not asking questions about all of the evolutionary processes that we know to be important. And so with that, I go forward. and thank all of the people in my lab, my collaborators, and Tanya who collected a lot of these samples, as well as funding agencies. And I'm happy to take a question if there